we're going to talk about residue control and breakdown. So it's titled Results from the Field, and I'll do some of that, but really I like to talk about really the science behind it, because again, there's so much that's out there for managing residue and soil health and microbes. So I always like to explain what to look for in products and try to be more informative, and then just show what a product like ours can do. Now, I want to just start with a thought, and this is something that really has come to us over the last year or so, working with customers that are using a diverse biology like our Bio 800 Plus line. That historically, we stopped tilling to improve our soil. But what we're seeing now, and just kind of bear this in mind as we go through the talk, and I'll summarize with something like this, that today when we see that when we improve our soil, that actually helps enable us to reduce tillage. More on that to come. So we'll talk about crop residue management, with the role that microbes play, and what to expect when you use a product that really can do the job. So first of all, you all know that your crop residue has a lot of value. You can leave it on the field, which again helps protect the soil from erosion, moisture retention. There's a lot of carbon and food out there that will help build the soil. Or you can pull it off because your residue also has value as feed, maybe as an energy feedstock, or other things that you could do with it. So you've got a choice. But if you choose to leave it out there, again, we've got, well, regardless of your choice, different things can happen. So first of all, we know that if there's too little residue and the ground is bare, that can really lead to erosion. Erosion is one of the biggest sources of nutrient loss in your field. So we really want to minimize the erosion. And of course, you see that ugly picture on the right with crusting, and I've heard all kinds of stories, and I'm sure you all have seen what happens when you've got too little organic matter on top protecting the top of the ground. But on the flip side, if we have too much residue, like in the lower left-hand corner of that photo, that we can see things like, of course, the equipment can jam, it can be a mess to try to plant through, but the soil can warm unevenly, which can lead to uneven emergence. And there's some really good data out there that shows that even a few days difference in emergence can result in pretty significant hits at the end of the season in yield. So we want to make sure we manage the residue properly. So, and last, fertilizer is expensive. Yeah, it's come down a little bit, but it's still a pretty meaningful part of the cost of production of most crops. You've already paid for the nutrients in that residue, and we want to get it out of the residue and back in the ground for your future crops. So if we just look at corn, for example, for every 40 bushels of corn, there's about a ton of residue. So your 200 bushel an acre corn crop is producing 5 tons of residue. In each ton is about 17 pounds of nitrogen, 4 pounds of phosphorus, 34 pounds of potassium. And you'll see, potassium is pretty high also in soy and wheat, because potassium is a nice component that helps with stalk and stem strength. And there's also some sulfur. So again, you've got a lot of nutrients out there that we want to unlock, but not a single bit of the nutrients that you've paid for in that residue will ever become available unless that residue is degraded. And you all know that. So what are the factors that influence decomposition rates? Well, the only thing, and it's a process called mineralization, that can take organic material like a corn stalk, soybean stems, etc., and break it down to the basic elements that are nutrients that the plant root can take up. The only things that do that are microbes. So really the better question is, what are the factors that influence microbial activity? Now there's a lot, and one kind of an obvious one is Do you even have the right microbial populations out in the field to begin with? But otherwise, we can simplify this. The two big ones are going to be temperature and moisture. Generally speaking, if it's warm and wet enough for your crop to grow, I don't care if that's a cover crop, a weed, or the actual crop you've got microbial activity. If it's too cold, too dry, and nothing's growing, then the microbes are probably just dormant. Now, there could be moisture in the ground, and they find the moisture areas between soil particles and such, so they still may be doing some things in the soil. But generally, we need the right microbes, we need some temperature, and we need some moisture. So I keep talking about microbes, and here's a list, and we're going to talk about each one because it's important when you're looking at different microbial products to ask a few questions about what's in there, and you can't always tell by the label, for various reasons. So here's a list of the types, the big buckets of microbes, from the most abundant bacteria. They're the smallest, and with actinomycetes, the fastest to grow. You can see there can be up to 1 billion in just a gram of soil. That's less than a teaspoon of bacteria, and you can have 2 tons in terms of the biomass. And as we go down the list, they're larger, they're less plentiful, they're slower to grow. Fungi, protozoa, nematodes. The fungi in particular, you can see in number a lot less, but in weight, they're much, much larger in a healthy soil because fungi are a lot larger than bacteria and actinomycetes. So are protozoa and amoeba. But look at the ranges, especially fungi, from nothing, 9 pounds, to over 13,000 pounds. This is important because we're going to talk a lot about fungi. So these are the different microbe types. So as I go through this presentation, we're going to talk about what they do and what to look for. And, I say that they're all necessary to make your soil really work at the top efficiency. And I say the way God designed it to happen. And I say that intentionally. I say that intentionally because, a little bit about me, I have a doctorate in molecular biology. I earned that doctorate a long time ago, it was in the 1980s. Back then, I can tell you, even though I had a Christian faith and I had no issues with that, and I did well in school, which means I regurgitated what I was taught, which was millions and millions of years of random evolution. And through time, I actually have used my education, dug into the raw data, and looked at the probabilities and such, and asked myself, okay, so can random processes come up with the elegant complexity? And it's mind-boggling. And the answer is no, because we see absolutely no example of information being created. It gets rearranged and destroyed. So the mathematical term for the long evolution is absurdly impossible. So if you want to know more about that, I have a lot of passion because I was fooled. And really what the data shows now, that I am humbled and grateful to say, is the best explanation is found in Genesis chapter 1. And I mean that as a scientist. So again, that's a whole nother talk. So let's get back to this. So bacteria and actinomycetes. You can see that top picture, kind of the greenish background. That's a picture of bacteria. They don't all look like that. They come in different shapes. Actinomycetes in the lower corner, that's a close-up. They're really tiny like bacteria, a tad larger. Again, really important in the soil. They can degrade organic matter, so they do play a role in breaking down your crop residue. They're really good at grabbing nutrients, because if we remember from a number standpoint, there can be over 1 billion in just a gram of soil, over 4 tons by weight of each time in a good soil. It can also be 10 times lower than that in a bad soil. They immobilize nutrients because they tend to live within half an inch of a living root. They dominate tilled ground, and that's important, and we'll talk about this, why that matters. And again, if you've heard me talk before, I talk about carbon to nitrogen ratios. This is not only important in the nutrient cycling talks I give about nutrient use efficiency, but really also very important when we talk about crop residue breakdown. So bacteria and actinomycetes, for every nitrogen, have about 5 carbons. As a biologist, that is an extremely high nitrogen requirement. They suck it up, they grab onto it, they tie it up. Most microbial products are just bacteria. And it's because they're those are the fungi that put out the long hyphae that intertwine to form mycelia. These are the kinds of microbes that are most efficient at breaking down organic matter, woody material, and we'll talk about what that is. So fungi are the best degraders out of all the soil microbes. Fungi eat bacteria. They're really good at unlocking nutrients like phosphorus. Because they're so large, they actually add carbon to the soil. They need oxygen. Now, fungi live longer than bacteria, but they're wimpier. So when we till soil, we disrupt the stable environment they need. We chop up all that white stuff, basically. We knock them way back. We expose more food for the bacteria. They grow much faster than anything else. They eat the food, end up with a bacteria-dominant soil. Now, fungi, we go back to carbon to nitrogen ratio, they're in a better spot than bacteria. Instead of 5 to 1, it's 15 to 1. So 3 times lower requirement for nitrogen. Very few microbial products will contain fungi. They may contain 1 or 2 species because, again, they require oxygen. They really don't form spores that you can store for a long time like some kinds of bacteria. Now, the protozoa, amoeba, and even nematodes. Now, these are predators. Now, remember, we're going from, 1 billion or more bacteria in a gram of soil to now very few of these because they're a lot larger and they help keep things in balance. These are predators. They eat everything. They eat each other. A single protozoa or amoeba can eat 10,000 bacteria a day. So we don't need 1 billion protozoa. There's not enough food. We can have far fewer because they're keeping the bacteria in check. Most important, look at their carbon to nitrogen ratio. It's not 5 carbons for 1 nitrogen, it's 30. It's 6 times lower. They also eat fungi. So this is really important, like protozoa, amoeba, and nematodes. And that's a picture of a predatory nematode eating a soybean cyst nematode. A lot of nematodes in the soil do not eat your plant roots, they eat everything else. So we want them there. These all need oxygen. But look, we got a protozoa eating 10,000 bacteria. That bacteria has a really high nitrogen content. To a protozoa or amoeba, that level of nitrogen is toxic. It's too much. They have to get rid of it, just like you and I do if we eat a really big protein-rich diet. Not to be indelicate, urea and urine sound similar. It's not an accident. That's us or animals getting rid of excess nitrogen because we're also about 30 to 1. So what do? They spit it out in the form of ammonia and nitrate. What are the 2 forms your plant roots take up the most readily? Nitrate and ammonia. Where? Well, the bacteria live within ½ an inch of a root. It's happening right in the root zone. So we complete the cycle. This is why we want all the different microbial types to really efficiently, again, the way God designed things to work, to keep things cycling back and forth. By the way, almost no microbial products will contain nematodes or protozoa or amoeba. Again, These are, you can't dry them and keep them viable. They do not form spores. So if you're being sold a product that's a powder, it may still be a good product, but it's not going to contain a lot of fungi and it won't contain any of these. If it's a liquid, it's tightly sealed at room temperature on a shelf, it cannot contain these either, because these, like fungi, need oxygen. So really, the only place you're going to find these will be in a compost tea type product, something even that would be refrigerated. So now we've talked about carbon to nitrogen ratios. So we're asking microbes to break down our crop residues. And so remember, I'm a molecular biologist, so all this stuff on the right I love. Don't worry, there's no quiz at the end, and I have a point for doing this. So when you look at, what I'm showing you is cellulose, lignin, and hemicellulose, three of the most abundant natural polymers on the planet. This is what comprises most of your crop residue, these three molecules. So let's start with cellulose. First of all, when we look at these structures, the O's are oxygen, the H is hydrogen, the C's where we show them are carbon, and you see all those lines that come together in points? Those are lazy. Oh, that's scientists. We're lazy. Those are carbons with hydrogens. We just get tired, so we don't write them all. So you look at all these things, it's C, H, and O. So if we start at top, with cellulose, that kind of triangular or weird looking shape that goes up and down, that's sugar. That's what a sugar molecule looks like a glucose, which is part of sucrose, table sugar. Cellulose is just a really, really long, straight chain of sugar. These straight chains can pack tightly together, and when they do, they form those rigid bars you see in that picture, called microfibril. I equate these to the rebar in concrete. Lignin doesn't have a set structure. You look below it, it's amorphous, it goes all over the place, it's incredibly strong, it repels water, it's the concrete. So we're looking at cellulose rebar surrounded by a lignin concrete. Hemicellulose is kind of a glue. So again, when we look at all this, I see C's, H's, and O's, but where's the N? N is nitrogen. There isn't any. So we're asking microbes that have certain nitrogen requirements to eat crop residue that's basically devoid of nitrogen. So what happens? So let's look at the carbon to nitrogen ratio of different crop residues. Alfalfa, clover, and such, no problem, plenty of nitrogen. Now let's go to corn. 1 nitrogen for 55 to 60 carbons. Wheat and other small grains, it's 1 to 80. So we're asking, if you don't use any microbial product and you till your ground, you have mostly bacteria out there. We have a lot of data on this. You have a very bacteria dominant field. So we're asking bacteria to degrade crop residue that has almost no nitrogen in it. So what are they going to do? They're going to steal it from the soil. So what happens? In the spring, the corn comes up, it's yellow. You do a soil test, because you probably put nitrogen down. Nitrogen is in there. If your soil test is somewhat sophisticated, it may even tell you it's organic nitrogen. What does that mean? It's in the bacteria. The bacteria are eating it. They're tying it up because they're spending so much energy trying to degrade this low carbon corn or wheat. So we need to have the right biology to get this job done. So what are some of the different products that are available to help us break down residue? Now there's equipment that will chop and crush, and that's good. Because if you think about your skin, you usually don't have an infection unless you get a cut. That's an entry point for a microbe to come in and cause the infection. Same kind of thing with your crop residue. The more you cut it or crush it, it creates more entry points for microbes to go in. So we've got more activity across the residue. It's just going to break down faster. Now, some products are just sugars. They say, oh, you got all the microbes you need out there, just feed them, give them a blast of sugar. Now, hang on. If you have all the microbes out there, they're already trying to feast on a carbohydrate-rich diet called your crop residue. They don't need more sugar. They need nitrogen. So there are a lot of different recommendations that say in the fall, if you've got a residue problem, put down nitrogen. Logically, given everything I've said, this makes sense. However, I can tell you there's a really good study out of Iowa State University where they looked at this, and kind of surprising to me, but again, the data is good. Nitrogen alone generally is not sufficient to make any difference in how the residue is broken down. I suspect why that's true is they're looking at most fields that probably have some level of tillage, and I've even seen no-till fields where we go out and we do a test. They're still so bacteria dominant, there's just not the fungi out there to break it down. So again, we can spread the feast for the microbes, but if we forget to invite the guests, which in this case are the right kinds of fungi, we've done nothing. The food's going to sit there. So there's also, of course, microbial products, and there's a number of them out there. So this is a comparison. And look, I'm not going to name different products or companies by name. That's not my place. I don't like it. I suspect you don't either. That's not what I'm trying to do. So what I've just taken is our product is Bio 800 Plus Breakdown. And I've got 7 different products that I went out that I get asked about a lot. I see advertised a lot. I'm just calling them A through G. So I'm again, not trying to pick on anyone's product nor declare if they work or not. That's their business. Our product is very biologically rich, and we have the right fungi in there, and I'll talk more about that later. But then I look at everything else, A to G. Bacteria, by the way, bacilli, Bacillus pseudomonas, those are bacteria. Except product B, nobody's selling fungi. By the way, yeast is a fungus, but yeast are not degraders. That's the wrong kind of fungus. Product B is interesting. So that one, it says about 70 bacteria and fungi. I can't find what they are, but if they've got the right kind of fungi and they're like the penicilliums and aspergilli, et cetera, then that could be a good product. I just don't know, but that could be. But for the most part, this disappointed me. So especially, let's look at what really happens in the field now. So look at the picture in the lower right-hand corner. We've all seen this. You've got next year's crop growing on a rotation, or last year was corn. And what do you see out in the field? Chopped up corn stalks. There they are. They're still there all over the place. Why? Well, let's look at the cross section of corn stalk. By the way, this is going to be similar to other things like wheat straw and such. I just found a good picture of corn. That middle part that's almost white, really light brown, called the pith, that's the largest in mass, but it's really low density. It has a good water content. It's only about 11 to 12% of the weight, even though it's a big part of the volume. Now you look at all those kind of brownish black dots all throughout the pith and all around the outer rim. Those are the vascular bundles. Those are the tubes that water and nutrients flow up and down the plant through. Now the rind is that outside part. Now it has some vascular bundles, but that rind, even though it's not much by volume, it's 75% of the weight of a corn stalk. It's triple the density of the pith. Furthermore, the outside of it is coated in wax. That's to help keep water inside your corn stalk. It's a protective layer. That rind is also really high in lignin and in cellulose. Remember, lignin and cellulose, concrete and rebar, they're extremely strong because they repel water. They're really hard to break down. So that's why you see the rinds, because it's that cellulose and lignin that's hard to degrade. So now let's look. This is a picture again from one of the products I showed before, and I'm not going to say which one. And this is from the website advertising this product that is supposed to break down residue. Look what happened. The pith is gone. Well, the pith is gone anyhow. There's nothing in it. It's just watery, it's easy to digest, but we can see now the vascular bundles and that outer ring, the rind, not much going on. Now, this is our product where it's very fungal heavy. So the way we make our product, we are brewing almost a designer engineered compost tea. We're constantly changing how we brew it, adding different foodstuffs, pH, temperature. We can, in our process, enrich for different families of microbes. When we make breakdown, we enrich for fungi that are really good at degrading cellulose and lignin. So this is what we see. Look, you apply the product, and remember, these things, because it's a waxy outer layer, they're going to degrade from the inside out. So like you see in the left-hand picture, go and see how well that stalk crumbles or the wheat straw breaks apart, and then look for the black or the white fuzzy and even blue fuzzy I see there in the right-hand picture toward the top. That's fungi doing the job. That's what you want to see. So again, our product, we've got all different kinds of microbes, the bacteria, the fungi, protozoa, amoeba, because again, we want to cycle all these nutrients. We don't want them to stay in the fungi or in any one form. We want them always cycling back into the ground for your crop. We do put a sugar source in just to give them a boost, but we also put a little bit of nitrogen because even fungi can use a little bit of nitrogen. It's not certainly going to contribute anything to your field. This is just to help the fungi have a little bit of a nitrogen source to break down the residue. So how we apply it, whether you're using our spring product, and in that case, we're tweaking the microbial populations for those microbes that really help with germination, rooting, and nutrient cycling, improving nutrient use efficiency. That's 1/2 gallon an acre. You can apply that in the furrow, over the top, etc. Our residue breakdown product is also 1/2 gallon an acre. It's a little bit different because it's fungal dominant. We put a little nitrogen in. And of course, since we're breaking down residue, we want you to spray it over the top. We like you to use 15 gallons an acre of water to wet it into the residue mat. Depending on if it's already wet out there, you could probably use 10 gallons an acre. Use it as close to harvest as practical. And this is important because remember, we need temperature and moisture. And I'll give you, now here's an example. This is from Iowa. This is last fall. We applied the breakdown to this in October. 3 weeks later, we took this picture. This is November 10th. All the farmer did, this is the different parts of the same field. He vertically tilled. So he helped chop up the residue, which is good. He didn't work it in, he didn't chisel plow or anything like that. On part of the field, he applied the breakdown. Look at the difference. Now I have to stress, we had really good temperature and moisture. This is a good result. So to be fully transparent, if it's dry and cold, you won't see residue breaking down this fast. However, whatever the conditions, go out, touch the residue, see how it breaks apart, look inside. What this particular grower said is because of the breakdown, he wasn't going to have to chisel plow. So we're putting good, healthy microbes on the soil. Now we help enable no-till. This is in Minnesota. So here's another example. So I keep saying temperature and moisture. So for example, if you have a really late harvest and you're in a place where it's going to be winter or winter already arrived. So let's pretend you're in Iowa and it's after Thanksgiving and now the ground is below 40 degrees, snow is coming or maybe is already there, or you're in a different part of the country where it's just bone dry and you're not even getting morning dew in the residue mat, then you're not going to see a whole lot of breakdown. And certainly if it's that late in the season, don't spend your money. Just don't do it because it's not worth it. Wait until spring, like this grower did in Southwest Minnesota. Here he applied it pre-plant, and again, after a couple weeks, corn stalks are breaking down, He's got another field with wheat straw that's breaking down, and you can just see the fungi growing in it. So it really depends on where you are and what you think the temperatures and the moisture profile is going to be if you can apply. Generally, October is really safe. November, depending where you are. December, most parts of the country, I'd wait till spring. This is different. This is golf. This is a PGA course in Las Vegas. Thatch is a problem in golf and athletic turf. What is thatch? Well, it's basically crop residue. It's the dead roots and clippings and all that other stuff. And what we're seeing in golf and athletic turf is the same thing in crops, where basically the microbes are eating the thatch layer. So you don't have to worry about dethatching. Just another little testimonial. Another benefit is if you're putting down the breakdown product and doing a cover crop or a winter crop like winter wheat, and if you have enough temperature and moisture to germinate a seed, you have enough temperature and moisture for the microbes to do their job. Another benefit is the microbes will help with that germination and stand. So, the corn seed there on the left, that was planted in 42-degree soil, which is kind of nuts, and with a early spring applied product, and you can see already some germination. Here's another example. Now these are wheat beans. So these are soybeans following a winter wheat crop in the Boot Heel of Missouri. Again, trying to break down that wheat residue, because by harvest time, it's typically still there and causing problems. This is a first. The field was so wet, and thank God there was finally moisture down in Southwest Missouri, because last year was terrible. They flew it on with an airplane at 5 gallons an acre. And looks what it's doing to the soybean crop that was planted 10 days later when they could finally get out in the field. Again, there's so much moisture. Microbes move with the moisture. So if it's really, really wet, you don't need to use 15 gallons an acre. But again, the microbes are not only getting to work on the residue, they're helping with the germination and the root mass. And again, the picture says it all. So I spoke a little bit earlier about how we treat the soil well, we get good biological activity, and that helps enable no-till. So I'm not going to go into a lot of the different science here. I did it a couple weeks ago in a webinar I did with Crop Life, and I'm going to do this going forward in other webinars. But we are told all the time how customers say, wow, the ground is so easy to work. We don't see clods. We've got an open soil. We actually get water infiltration. And again, good water holding. What's going on? Well, again, this is good biology that's taking soil that can be compacted and instead creating macro aggregates from the compacted soil that doesn't have it. So the punchline, what creates the macro aggregates that open up the soil, get rid of clods and all? It's fungi again. So I go back to the importance of fungi, the microbe family type that tends to be missing from most farm fields. Fungi working with root exudates produces a glue, and that glue binds soil particles together to make those macro aggregates, which really gives us that nice soil texture. The fungal hyphae, all that white feathery stuff, encases those macro aggregates, as will plant roots and root hairs. Now it's a constant fight between bacteria and fungi, because that glue, bacteria love it. So when we till fields, now the glue is exposed to the bacteria, they eat it, we have to start all over again. But if we can keep the fungi and the glue out there, then we actually improve the texture of the soil. So our recommendations, remember, to break down crop residue, you need microbes. And for microbes to work, really the best ones to break down that cellulose and lignin, which that's what gives trees its strength, wood the strength, your corn stalk strength, etc. We really need fungi. And we need not unicellular fungi like yeast, but the ones that produce those white feathery mycelia. Apply when it's not too late in the season. If it's bone dry, or if it's winter, don't spend your money. Just wait till spring. But if you can get it on early, that's great. Even though a lot of people use nitrogen, logically, fall nitrogen should help microbes break down residue. Iowa State has good data that says no. And again, I think it's probably because the right microbes weren't out there. The more you can do to crush and chop your residue, the better. That's more entry points for the microbes. People ask me all the time, so should I work the residue into the soil? Now, on the positive side is the more soil contact, the more chances we're going to have, if you're not using a degradation product, or even if you are, that you're going to get fungi from the soil, assuming you've got decent fungi in your soil. But the bad side is, again, tilling sets back the fungi. The bacteria eat the glue. It can lead to erosion. So really, when you use a product like ours, no, you don't have to work it into the soil. A vertical tillage pass, if you need to, seems to be okay, but otherwise, if you just got equipment that does a good job crushing and chopping, you should be fine. So Ritu, with that, that's, crop residue control 101. And if there are questions, feel free to enter them, and I would be happy to answer them. If you want to think about it, that's my email address, dstark@holganix.com. Feel free to email me. I do a lot of webinars. I get a lot of emailed questions. So you're not bugging me. This is what I love to do. So thanks for your attention. Ritu, do you